many patients question, especially with respect to a phase 1 clinical trial, is will I be a guinea pig? I mean, this is a trial that's considered first in human. So what does that really mean? Is there research that's already been done before? Is there any background information that I might be able to access to try to figure out if this trial might actually help me or not?
Absolutely. This is a very important question, and is one of the most common questions that we get asked as early phase clinical trialists in our clinic. So some of the things that I'd like to say is that all phase 1 trials or first in human trials should have shown some evidence or benefit or activity in the preclinical setting. So they have in animal models or cell lines. So they have to meet a certain standard before they're taken into the clinic. The next step after the standard is met is to conduct a phase 1 trial, where, as I mentioned earlier, the primary objective is to assess the dose and the safety of the drug. Usually it's often hard to project if a drug that has shown promising activity in a preclinical setting will have the same degree of activity in humans. But that is why we conduct a phase 1 trial or a first in human trial to investigate all those questions. Generally speaking, I would say clinical trialists work with the goal and optimism that these phase 1 studies will be safe, and at the same time, as a secondary objective, show promising activity against the patient's tumor so that the patient also benefits from this novel agent or novel modality that is being tested. So as a consensus, I would say clinical trialists say, whenever we get asked about this question, if a patient feels that we are testing a drug in them, it is true, we are testing a drug in an individual when we are talking about a phase 1 trial, and it's the first time that the drug is being tested. But for that test to happen, before that, there's a great deal of work that has gone into testing the drug in a preclinical setting to see if it would actually have some benefit. But to make projections based on this preclinical setting, it is difficult. So that is why the phase 1 trial is being conducted.
I'll add to that and say that sometimes you'll hear things in the news that have sort of gotten ahead of even a phase 1 clinical trial. And so you'll hear, I remember, I think it was last year, I heard that blueberries cure cervical cancer. And I thought, well, that's not, that seems suspicious. And so I go and read the news articles, and it's, blueberries have been found helpful in a mouse model or in human cells. But what that means is that potentially promising treatment has not made it to the phase 1 trial. And it's what's amazing is how many promising therapeutics in a mouse model or in human cell lines don't even make it to the phase 1 trial. So you really have to have a very good foundation of preclinical data to even move it into humans. And I always try to emphasize that for patients, that it's not just like one trial in one mouse that blueberries found. To move it into the phase 1 trial level, you need to have preponderance of preclinical data.
Absolutely, and I would just add to that, when as clinical trialists we write a concept or write a protocol, we usually have to show evidence of a certain agent being active in multiple different cell lines or multiple different animal models, different mouse models, for example, or with different types of tumors. Or different cell lines of the same tumor to show that that agent has activity before we propose a trial in humans based on that agent. So completely agree with what Dr. Chino just mentioned.
Well, thank you. That's very helpful to hear about the preclinical research, because that makes a phase 1 clinical trial seem a little less scary, knowing that there is research that's already done.